Let me start by welcoming you all to this webinar. Thank you all for joining and being here with us today. So it is so wonderful to see so many of you joining for this nice webinar that we have prepared for you after the relaxing summer holidays. I hope you all had. So for today's webinar, before we get started, I would like to give you some general information. Let me start with that 1 second. Yes, so by default, your video will be off and your audio will be muted. This webinar is being recorded and will be later available on demand. So by participating in this webinar, you consent for this session to being recorded. There will be a Q&A session available after each speaker presentation. And we invite you to write the questions in the chat. So we will have 2 modes. So the moderator will read the questions to the speakers. But if you feel brave enough, also you can also use the tool here in Zoom to raise your hand and you can formulate the question yourself. And at the end of the webinar, there will be a short survey. We would appreciate if you take some seconds to give us some feedback on that. So a little about the agenda of today. We will start with a little introduction to automated patch clamp by Ali. Then we will move on with the presentation, the first presentation of today by Dr. Samantha Salvage. And last but not least, we will have the presentation by Johnathan Silva. So I think without further ado, we can get started with the webinar. So let me introduce you to my colleague, Ali Oberglusberger. She has been with us at Nanion for over 17 years, and she's currently the Scientific Communication Manager at Nanion. She will be today's host and moderator of the webinar. And when you're ready, Ali, the floor is yours.
Well, thanks very much, Jamar, for this nice introduction. Yes, 17 years, it's quite a long time, isn't it? But welcome everyone to this webinar this afternoon, or morning, I guess, depending on where you are, entitled Exploring the Effects of Antibodies and Anti-Arrhythmic Drugs on Ion Channels Using Automated Patch Clamp. So as Jamar already mentioned, we're going to start off today's webinar with Dr. Samantha Salvage from the University of Cambridge, and then after Sam's talk, we'll have a short Q&A session, and then we'll hear from Professor John Silva from Washington University in St. Louis. But before all that, I'm just going to give you a brief introduction into the world of automated patch clamp, and in particular, the patch liner, just to make sure that we're all on the same page. I'm sure the people who are listening here, many of you are familiar with or perhaps using manual patch clamp and maybe even also automated patch clamp. But for those of you who are not familiar with the technique of automated patch clamp, I'm just going to go through quickly how it differs from manual patch clamp and how it works. So the technique of conventional patch clamp involves bringing a small glass pipette with a tip diameter of about 1 micrometer to the cell surface and forming a tight seal in the order of a gigaohm or more between the plasma membrane and the pipette tip. At which point, the patch of membrane stuck in the patch clamp pipette can be broken to access ion channel activity occurring across the whole of the cell membrane. I'm sure many of you can identify with this person here who's looking down a microscope, because they need to be able to see what they're doing, to be able to position the pipette on top of the cell. They also need an anti-vibration table, a Faraday cage. They need to be able to pull pipettes, and they need to be very skilled to perform the experiments. And although the technique of conventional patch clamp is a very information-rich technique and is still considered to be the gold standard for recording ion channels, conventional patch clamp does have its limitations, some significant limitations, including its low throughput and the requirement for skilled personnel to perform the experiments. And it can become quite a bottleneck in some projects requiring electrophysiology data. Planar patch clamp, on the other hand, doesn't involve pipettes, but rather involves a planar substrate. And although it was first described in the 1970s by Kostyuk and others, they used a polymer substrate and very large holes in the order of 40 micrometers or more, and very large snail cells, not mammalian cells. In the 1990s, it became apparent that there was a need for higher throughput patch clamp on mammalian cells, and companies such as Nanion sought to come up with a planar chip that could be used to record ion channels from mammalian cells in an easier and higher throughput manner. Nanion's automated patch clamp instruments use a planar glass layer made of borosilicate glass. So it's the same glass that you would use to make your patch clamp electrodes from. There's a small hole in the middle, and cells in suspension are attracted to the hole using suction from underneath. This means that there's no need for microscopes. You don't need to be able to see what you're doing. There's no need for a anti-vibration table or a Faraday cage, and you don't need to pull pipettes. So the whole thing gets a lot smaller, it gets a lot easier to use, and there is also the option to record from multiple cells simultaneously, thus increasing throughput. So using automated patch clamp, we can record in different configurations. In the on-cell mode, the cell is attracted to the hole. A good seal is made between the cell membrane and the glass, but the cell is not yet ruptured, or the membrane is not yet ruptured. And this means that we can record these ion channels here, which are stuck. Here we go. These ion channels here that are stuck in the hole. And there we see openings of, these stepwise openings of 1, 2, or in this case, 3 ion channels opening and closing. If we then rupture the patch of membrane that's stuck in the hole, we get access to ion channel activity occurring across the whole of the cell membrane. And this means that when we use a stimulus, this stimulus may be a change in voltage, it could be the addition of a ligand, or some kind of other physical stimulus such as heat or pressure. We get activation of multiple ion channels simultaneously. These then desensitize or inactivate, and then there's channel closing as the stimulus is removed. There is also a third recording configuration that we can use with automated patch clamp, which is not illustrated here, but this is the perforated patch technique. So in this case, we use a perforator, usually an antibiotic, in the internal solution, which makes pores in the membrane, so we get access to ion channel activity on the whole of the cell membrane, but the internal solution remains intact. So there are 4 products for automated patch clamp from Nanion, ranging from the Porta Patch and the Porta Patch Mini, with add-ons for external and internal perfusion, for temperature control and light activation, for example. There's then the patch liner, a fully automated system for recording 4 or 8 cells simultaneously. And last but not least, the SynchroPatch 384 for recording up to 384 cells at a time. So for the purposes of today's webinar, we're going to focus on my personal favorite Nanion instrument, the patch liner. So the patch liner is a fully automated patch clamp system for recording up to 8 cells in parallel. It uses patch clamp amplifiers that you might be familiar with already from conventional patch clamp recordings, the Hika EPC10 amplifiers, and with the add-ons such as the Dynamite 8 for automated dynamic clamp recordings, and a sophisticated temperature control, it really does expand the experimental possibilities. The chip on the patch liner is the MPC16 chip. This, like all of the chips from Nanion, has a planar borosilicate glass layer with a hole in each well. But with the patch liner chip, it's sandwiched between 2 plastic molds, making microfluidic channels. And this means that exchange of the solution is very fast. And it's really the flexibility and the quality of recordings that you get with the patch liner, coupled with its higher throughput compared with manual patch clamp, which really makes it perfect for academic labs. But before we hear from our 2 academic labs that are using the patch liner, I'm just going to make a blatant plug for the SynchroPatch 384 instrument grant that we have running at the moment. So the winning proposal is going to receive a SynchroPatch 384 for 9 months in the lab, including consumables and assay development support. And this is completely free of charge. So we are going to be accepting applications soon. And the deadline for applications is going to be some time around the end of October. So please think about a proposal and think about putting in a proposal for that. So I won't bore you anymore. I'd like to introduce our first speaker now, Dr. Sam Salvage from the University of Cambridge. Sam completed her PhD on the effects of intracellular calcium and calcineurin on gap junction resistance and its implications for cardiac action potential conduction at the University of Surrey. She then investigated the effects of cardiac ryanodine receptor dysfunction on conduction and arrhythmiogenesis in the Department of Physiology, Development and Neuroscience at Cambridge, and ryanodine receptor gating properties with Professor Angela Dulhunty on a visiting fellowship at the Australian National University. She currently studies the structural and functional regulation of cardiac NAB1.5, in particular by beta subunits and intracellular calcium in the Department of Biochemistry at the University of Cambridge, working with Dr. Tony Jackson and Professor Chris Wang. Sam's been using the patch liner for about the last couple of years and is going to talk to us today about single chain antibodies and their effects on voltage gated sodium channels. So thanks, Sam. I will stop sharing.
I'd like to start by saying thank you to Ali and Shaymar and the team at Nanion for the opportunity to talk about our work here today. I'll be focusing on a recent endeavour in our lab where we're using single-chain antibodies in order to target voltage-gated sodium channels and then hopefully observe functional effects of these single-chain antibodies via planar patch lamp. So voltage-gated sodium channels comprise a family of 9 isoforms. They're typically found on excitable cells, such as the heart and in the brain, where they control the influx of sodium ions in a voltage and time-dependent manner, which is crucial to the action potential. They comprise one long single polypeptide organized into 4 domains, domains 1, 2, 3, and 4, each consisting of a voltage sensing domain, segments 1 to 4, and a pore domain, segments 5 and 6. All of these are tethered together via extracellular and intracellular loops. In their 3D structure, they're organized into this pseudo-tetrameric organization. I've highlighted domain 1 in purple, domain 2 in turquoise, 3 in yellow, and 4 in gray. The voltage sensing regions sit at the periphery and the pore domains in the center, creating the central ion cavity through which sodium current flows. They typically exist in vivo in complex with 1 or more regulatory beta subunits. And our primary interests are concerned with NAB1.5 and NAB1.7, given their function in cardiac arrhythmia and pain respectively. We're also interested in how they're regulated by the beta subunits and the potential to selectively modulate their function. And one of the ways in which this can be achieved is via antibodies, which comprise one of the most rapidly growing classes of drugs. In particular, we're interested in single-chain variable fragment antibodies, or SCSVs. These comprise just the variable heavy and the variable light chain of a full IgG. The phage display library we have access to is completely human in origin and can be produced in vitro. The relatively smaller size can enable more precise or higher resolution binding and labeling, and also mitigates for potential immunogenic risks associated with FC recognition. The library we have access to is huge, with a complexity of more than 10 to the 13 clones. And although antibodies have had success in cancer and autoimmune diseases, to our knowledge, they're very limited or non-existent therapeutics for voltage-gated ion channels, in particular voltage-gated sodium channels. However, there are a number of antibodies in the literature that have been shown to have functional regulation, including against NAB1.5 or its neonatal splice variant. The first example shown here is a polyclonal antibody that has been generated against the neonatal peptide of the domain 1 S3 and S3S4 linker segment. This comprises the specific region in which alternative splicing occurs between the neonatal and adult form. Application of this antibody to this polyclonal antibody to both adult NAB1.5 and neonatal NAB1.5, even at low doses, shows specific inhibition of the neonatal sodium current, and at higher doses, a very marked reduction in this specific manner. The second example here is, again, a polyclonal antibody, Well, it's targeting the extracellular loop 3, which is the domain 1 S5 S6 loop. This E3 or extracellular loop 3 region represents a common point of sequence diversity across many ion channels. And here the polyclonal antibody again exhibited a reduction of peak sodium current. So we are essentially wondering if it would be possible to achieve functional regulation of sodium channels through a smaller single-chain variable fragment antibody. And I would like to point out even identifying high affinity and selective single-chain antibodies that bind can still be useful research and labeling tools. So to do this, we have set up a pipeline of work that is cohesive with the research and technical skills of our team. We do sequence alignments and structural alignments to look for unique epitopes. And once identifying those, we order the peptides and use them to select phages from the library. And this is essentially follows by a panning process, whereby the target protein or peptide is immobilized, exposed to the single-chain antibody library, and then undergoes stringent washing to remove non-binders, followed by an elution step, which releases the polyclonal population of phage binders. These are then expressed in E. coli, and following a series of purification steps, we end up with a population of clones that are monoclonal and targeting the peptide of interest. We then undertake a selection of a number of screening tests where we screen against the peptide against which it was generated, as well as the full protein expressed in stable cell lines. And then the most promising are taken forward for functional analysis. So I'm just going to go through this with two structural examples. The first one is the E3 region just mentioned from the prior study. This is the domain 1 S5 S6 loop. To see if we can get the same effect as what has previously been reported for a full IgG. And also another area where we have highlighted a unique sequence in NaV1.5 is in the domain 3 S3 S4 voltage sensor. So the first step after generating the population of different clones against the peptide of interest is to go through an ELISA screen. In a 96-well plate, we can test numerous clones. This graph has been simplified just for illustrative purposes, but essentially from 96 clones, we typically find in the order of 6 to 10 positive binders. Streptavidin has been included because it was used in the immobilization process of the peptide against which the single-chain antibody library was screened. So once we identify a few positive binders, we take them onto a cell impedance assay where the 96-well plate is coated with either the single-chain antibody of interest or BSA as a negative control. And then HEC293 cells stably expressing NaV1.5 are incubated on the plate, and cell impedance or cell index is recorded over time. An increase in signal here indicates that we're getting increased binding, so increased interaction of NaV1.5 with the single-chain antibody. And you can see, so here clone 2 is the most promising, and clone A6 for domain 3 S3 S4. But all of these clones are still showing very good signal. It'll be clone 2 and clone A6 moving forward. We also, to prove the specificity of binding, we repeat the experiment with the blocking peptides, the peptide against which the single-chain antibodies were raised. So if for domain 3 S3 S4, we incubate the cells on the single-chain antibody, in addition to increasing concentrations of this peptide, we find we abolish the interaction. So that's great. We have some positive clones, but then we wanted to test them for function using automated planar patch clamp. The first thing upon receiving the Nanion patch liner was that we wanted to be sure this does compare well with manual patch clamp. And at that time, we were working with NaV1.7 and looking at the effect of NaV1.7 with NaV1.7 beta 3. These are some representative activation traces whereby increase in step depolarizations elicit increasing currents and inactivation traces. You can see from the current voltage plots that the presence of beta-3 causes an increase of peak current. And then replicating that with the patch liner, we get very beautiful traces with very good voltage control. And we see the same effect, an increase of peak current. And when you directly compare the magnitude of those currents and the voltage sensitivity, it is completely reproducible. So that is incredibly encouraging. Another aspect of the patch liner is that it's great for rapid assessment of pharmacological agents. You can apply multiple doses or different agents in a relatively short space of time, which is great for small molecules where rapid dose responses are easily achievable. So for example, here, this is just one quick example of flecainide, which is a sodium channel inhibitor. We were able to apply this at 6 different doses, followed by a washout, with each dose less than a couple of minutes. And you can see this rapid effect. And indeed, within 30 to 40 minutes on a 4-channel system, we had a complete data set where you're seeing this characteristic inhibition. And plotting a hill curve to it, we get an IC50 well within the magnitude of what is reported in the literature. So that's great. We've also worked with small biologics, so toxins. These are small proteins, larger obviously than small molecules. When we perfuse this in, so this is protoxin 2, and it specifically inhibits NaV1.7. When we perfuse this in, we start to see an effect after about 10 minutes. So a bit longer than the small molecule, but well within the time course of the cell surviving in the whole cell configuration. And this is consistent with what we get on manual recordings as well, where we have to pre-incubate for 10 to 15 minutes. So we're going from a small molecule with less than 2 minutes to take effect, a slightly more complex small protein, 5 to 15 minutes. With a single chain antibody, this complexity is dramatically increased. It's 2 immunoglobulin domains. And we are also trying to target a region that might have some flexibility. So this is important in designing our patch liner experiment. So looking first at the E3 single chain antibody, we carried out these experiments on HEC NaV1.5 stable cell lines. We did the recordings in the whole cell voltage clamp configuration using medium resistant chips with 60% series resistance compensation. We carried this out so that we did the control recordings first and then perfused in the single chain antibody with the hope that we could get a dose response. So to do this, we used a fixed pulse protocol, which elicits a depolarizing step just to minus 25 millivolts, which is around peak current for NaV1.5. This repeats every 5 seconds for 30 pulses, covering a period of 2.5 minutes. Immediately after this, we apply an activating protocol in which we can get the IV curve data for, reapply the solution, and then move on to a repeat of the 2.5 minute minus 25 millivolt pulse every 5 seconds. Subsequent to this, the single chain antibody is applied first at 10 nanomolar, an incubation period, followed by the 2.5 minute protocol, an IV curve, reapplication of the single chain antibody, another 2.5 minutes, and then on to the next dose. And we had hoped to get to higher concentrations than this, but the number of cells that managed to persist through this length of protocol is quite few. But I will show you the IV curve. Looking just at 10 nanomolar, we really aren't seeing much effect at all, even with an N of 8. And then when you move on to 100 nanomolar, suddenly we've got quite a high attrition rate because of the amount of cells making it through, and still we're seeing no effect. So we're wondering, is this genuine, or is it that there's an insufficient concentration or an insufficient incubation time? So we considered one way to work around this was to pre-incubate our cells in the single chain antibody for an extended period of time. But that brings up the concern, will our control recording, if incubated at a similar period of time, be consistent with the currents that I've already showed you? So just to very quickly check this, we After dissociating the cells, we stored them in the fridge for 2 hours and then undertook the fixed pulse protocol with all 30 sweeps superimposed here, followed by the IV curve. And you can see this, the fixed pulse is completely superimposable, so very stable, and the activation traces show good voltage control and are typical of NaV 1.5 currents. And looking at the scale bar and capacitance values of the order of 10 to 12 picofarads, these currents are in the region of 5 to 600 picoamps of picofarad, which is completely consistent with the data I've already showed you. So we took the single-chain antibodies to the E3 region. We took clone 2 and clone 8, went to a concentration of 1 micromolar, incubated it for an hour, and then carried out these experiments. And even after this long an incubation at this higher concentration, we are seeing no effect, which was a little disappointing because we were hoping to replicate the prior work targeting the E3 region. There's a number of reasons why maybe we haven't replicated that. Our epitope may be slightly different, but also perhaps a bigger limitation is that in this case, the single-chain antibody being so small may not exhibit the same effect as the IgG, which could potentially be blocking the pore or occluding the pore in some way that reduces peak current. Maybe this is harder to achieve with the small footprint of a single-chain antibody. But we didn't give up. We moved on to domain 3, S3, S4 single-chain antibodies. Did a similar thing, used stable HEC NaV 1.5 cells. This time we went for a high dose, went to 1.8 micromolar straight away, fused it in a couple of times, and again used our fixed pulse protocol. But this time we decided to shorten the procedure slightly and try to just get more recordings. So it's the fixed pulse again for 30 pulses, 2.5 minutes under control condition. Then the single-chain antibody against domain 3, S3, S4 region is applied, incubated for a few minutes, and then the fixed pulse protocol starts. This first step covers the 5-minute period, the second one the 10, third 15 minutes, and 20 minutes, and so on. And between each of these, the solution is refreshed and an IV curve is recorded. You can see there isn't really much change, but there seems to be a trend towards decreasing currents towards the tail end of this protocol. But when you look at the IV curve and steady-state inactivation, there are no significant observations. So we went back to trying to pre-incubate the single-chain antibody for around an hour, and then try a washout in the same manner as the single-chain antibody was applied. So this washout is just applying the extracellular solution at the same time points. And here again, not really much happening, but the trend appears to be towards an increase in peak current. However, when you look at the IV curve and steady-state inactivation, not much is changing. But what we did notice is that this very first recording under the domain 3, S3, S4 single-chain antibody is showing a smaller magnitude of current. It's at least 50% smaller than what we're typically getting from that 1.5. So that made us wonder whether perhaps it was worth just trying different incubation times and recording control and the single-chain antibody only at that initial time point. And when we do this, the effect is actually much more stark. We can now see a noticeable decrease in peak current with the single-chain antibody, even at 30 to 40 minutes, with it not increasing that much more by 1 hour. Looking at the IV curve, there's a very significant decrease of this single-chain antibody without any effect on steady-state inactivation. So this is really quite encouraging. So in summary, the patch liner is great for assessing single-chain antibodies. We just have to approach the experiments in a slightly different manner and have a long pre-incubation step. So this means we need more cells in order to account for cell-to-cell variability, but the patch liner has a much higher throughput than we could achieve for this kind of work manually. Both the SCFVs I've shown here were able to bind to NaV 1.5, but only the domain 3 S3, S4 SCFV displayed any functional modulation. Interestingly, this S3, S4 motif appears to be a point of functional modulation with other polyclonal antibodies in the literature targeting the structural equivalent in other domains for different isoforms, including NAV 1.7. So we're also actually going to look, there's a unique peptide in the same region for NAV 1.7. It'd be nice to selectively target that. We will also then need to check that each single antibody doesn't target the opposing voltage-gated sodium channel. And in the future, we would also like to try and selectively target alpha subunits either in the presence or absence of beta subunits. But it's also important to point out that although this is looking incredibly promising, we do still have some further validation to be carried out. And we are aware of at least one report in the literature where antibody targeting NAV 1.7 was shown to have an inhibitory effect. And then another group tried to replicate the work and were not able to do so. So we want to be confident on this before going any further. I'd like to thank, in particular, Tony Jackson and Chris Huang, as well as Dr. Samir Hamaya, who is the in vitro phage display expert. I'd also like to thank the team at Nanion, particularly Nadine, Ali, and Christian, who helped us get off the ground with the patch liner. And thank you for your attention.
Great, thanks very much, Sam. Very nice talk. And yeah, you've been doing a lot with the patch liner. If anyone has any questions, please type them in the chat, or if you would like, also raise your hand, and if you feel brave enough, then you can unmute yourself and ask a question. We do have one from John Silva. He asks, I was really interested in the D3 S5 S4 S5 antibody. Why do you think there is no change in the inactivation curve?
Not entirely sure, but we are looking at this. So we are looking at it in NAV 1.5 cells without any beta subunits. So I guess maybe what you're alluding to is, could it be binding to a similar site as the beta 3 or beta 1 subunit? In which case we would expect to see a depolarizing shift of steady state inactivation. We aren't seeing that. Whether it would have any competing effects in the presence of beta 3, that is something we are hoping to look at. Sorry, I don't really know why it's not. But I understand why that would be interesting.
I see Marcus has his hand raised, so please unmute yourself, Marcus.
Hi, everybody. Really nice talk. Thanks for sharing the data. I have a question along with the D3, S3, S4 short chain antibody.
Mm-hmm.
Is this region intracellularly, or is the target out extracellularly?
It's extracellular.
Extracellular.
It's one of the extracellular loops.
And do you have any idea why, how the current could be inhibited? I mean, I think it goes in the same direction like the previous question. Did you check like surface expression reduced or so after the antibody?
No, we haven't checked that yet. So that is something we could look at. Although the turnover of voltage-gated sodium channels is in the order of days. I didn't imagine it would turn over in the period of time that we are recording, but it's definitely worth something to for us to look at. Whether it wasn't significant, but there was a slight shift in the voltage dependence of activation as well. But yeah, I mean, I can't imagine that the single antibody is directly occluding the pore. We essentially have quite a bit more work to do to delineate how the antibody is working.
Thanks a lot. Can I ask another question, a stupid one?
Of course, there are never any stupid questions, Marcus.
Sorry, but these short chain antibodies, are they also part of the human antibodies made in the immune system, or are they purely artificial?
So they are the variable heavy and the variable light fragments. They're the genes specifically encoding those regions, and we can actually in vitro put them into an IgG to make them a full length if we wanted to see if there were any different effects by having the entire footprint of the antibody.
Okay, but the human body always uses like the immune globulin, certain variants, and then you have in addition the variable parts of it, right?
Yeah, so the gene is from human, they're from human donors, but where they're made in vitro in bacteria.
Okay, I was just curious if there are any antibodies known that are these short chain ones in the human body. Okay.
Yeah, I'm not entirely sure.
Yeah, okay, that's fine. Thank you.
We did have a question come in about the impedance assay. They're asking, why did the impedance change when you added the antibodies? Is it somehow related to cell growth or viability?
So the antibodies are used to coat the plate, and the plates aren't cell binding plates. So if you add the cells in the absence of something that they can adhere to, you will get a low signal. If you've got a single chain antibody coating the plate, providing you attach cells that combine to that single chain antibody, you'll get an increase in signal. So although I didn't show it, if I had shown our hex cells that don't have NaV1.5, we wouldn't get an increased signal. And that's also why we don't get an increased signal with BSA, because the NaV1.5 cells can't adhere to the surface of the plate.
Okay, great, thanks.
But it does take a while for that signal, and that isn't necessarily indicative of the time course of the single chain antibody to interact with NaV1.5, because it also takes into account that the cells have to sediment.
Okay, thanks. I had maybe one question, one comment also. The medium chips, they're 2 to 2.5 mega ohms, right? You had giga ohms on your. Not that anyone's confused by that, you know.
No, yeah, very, very different.
I was wondering, so you're always doing multiple additions, whether you thought about doing like a single point addition and then incubating for longer.
Yeah, so as in you can apply differently to the different chips.
Yeah.
Yeah, that will likely be something we try to undertake in our further analysis. As I said, this is still quite early days, and we've got quite a few things to still look at. So I'll probably pick your brain about some of the different ways in which I can apply the patch liner for them.
Great. Okay, then I think we'll move on now. Thanks very much for a great talk, Sam.
Thank you.
And if anyone has any more questions, then please do write them in the chat. And I'm sure Sam will be still around a bit later to answer any questions that might come in for her as well. But we're going to move on now to our next speaker. I'm delighted to introduce now Professor John Silva from Washington University in St. Louis. John joined the Department of Biomedical Engineering at Washington University in St. Louis in July 2012 after receiving the Burroughs Wellcome Fund Career Award at the Scientific Interface. So John's lab, moving on to something a bit different now. So John's lab has created novel algorithms that predict whether patients will respond to class 1 antiarrhythmic molecules. These predictions are based on machine learning and approaches that leverage a deep understanding of drug interaction biophysics. His group has also developed software to provide a holographic display to physicians who perform catheter ablations for arrhythmia. And this software was recently tested in humans, and the results showed that physician accuracy was significantly improved with the display. John has now also co-founded a company, Centaur, to commercialize this technology. So thanks, John. We can see your screen. This is good. Over to you.
All right. Thanks. I really appreciate the opportunity to talk about the work we've been doing and share how we've been using the patch liner in our lab. Let's see. There we go. So a couple of disclosures. We do have a couple companies, but they're not really related to the work that I'll be presenting today. Before I start, I'd like to highlight the people who've really worked on this project. Zoltan Varga was a postdoc in the lab, and now he has his own lab at the University of Debrecen Medical School. Wandi Zhu did a lot of the biophysics work, and she is currently looking at different faculty positions. And then our clinical collaborators, Johnathan Moreno, Jennifer Silva, Sylvia Priori, and Lena Deng have also helped quite a bit with what I'm going to talk about today. So this is a classic figure that is shown to a lot of our students, and especially this says a lot to the biomedical engineering students that we teach. On the top here, you have the CAST arrhythmia trial, where the placebo actually was better for the patients than the drug. So survival was lower for patients who took a class 1 antiarrhythmic drug. And then shortly thereafter, in the mid-90s, the defibrillators were developed. And here you can see that the defibrillators helped quite a bit more compared to conventional therapy. So when I was a PhD student, this is kind of the dogma that we were taught, that antiarrhythmic drugs are bad and defibrillators are good. And so we should really just focus on device therapies. When I met my wife, who's a clinical electrophysiologist, I was talking to her about this, and she said to me that she actually prescribes antiarrhythmic drugs quite a bit. So a lot of patients are on antiarrhythmic drugs, and you don't really want them to be fully dependent on their device every time they have an arrhythmia to shock them out of it. You actually want to use a drug therapy to reduce the arrhythmia burden and only use that device when it's absolutely needed. Moreover, there's a lot of patients that respond really well to antiarrhythmic drugs. We just don't know exactly why some patients respond better than others. So this has become a bit of a theme for our lab and kind of what we've been thinking about. And so we started with the long QT type 3 syndrome, which is a fairly straightforward target in terms of understanding where the defects are and how someone might target those pharmacologically. So long QT type 3 is observed in patients who tend to have an increase in persistent late current. So after the sodium channels open to initiate the action potential, they usually close, and that's what allows the myocytes to repolarize and recover. If there's a defect in this inactivation, they don't close all the way after opening. That persistent influx in positive charges prolongs the ventricular action potential, and that's manifest on the ECG as a prolonged QT interval, which is why it's called long QT syndrome. So there are molecules that block the late current quite well. So lidocaine is one. It's used as a local anesthetic. It's actually a very potent antiarrhythmic drug. The problem is that it has to be given via IV. So the drug gets metabolized quite quickly, so it's only available to patients in the hospital. In the 70s, the mixilotine was developed, and it does not get metabolized as quickly. So it's available for patients to take orally. So they can get sent home with mixilotine. And you can see here that mixilotine is actually quite a potent late current blocker. So this makes sense that if you have a defect where you have extra late sodium current, if you give a drug that blocks that extra late sodium current, you should be able to address the pathology at its source. So this was data that Silvia Priori shared with us, which shows two different responses to mixilotine. Here's a patient that has a corrected QT interval of 480 milliseconds, and then it went to 420 milliseconds. So the patient responded quite well, going from a prolonged QT interval to what's considered a safe range below 450, about 450 milliseconds. A second patient that had even a more prolonged QT interval actually showed no response to mixilotine. And you can see some data from a lot of patients here on the right, where some patients didn't respond at all, and other patients responded quite favorably to the mixilotine therapy. So that was our goal, to understand why. There is some hints from classical work. So a lot of work has been done to understand the state dependence of sodium channel block. And there's an idea that these sodium channel blockers tend to bind the channel better when they are open or inactivated. So you have less binding here to the resting state. As the channel's open or inactivate, you get more drug binding. And that state dependence leads to use dependence as well. So you can have a certain amount of drug block when you just add the drug and leave the channel sitting there. And then you apply a single pulse. And then you can see as you apply repetitive pulses, you get more and more drug blocks. So this is relevant to the situation in the native heart where the heart's continuously beating. And you can imagine that the drug's going to be more effective at faster heart rates, or it's going to block more channels. So how did we get to the bottom of what's causing this variability? We have a method in the lab called voltage clamp fluorometry, where we introduce cysteines in 4 different constructs. We don't do these at the same time, we do them one at a time. We put a cysteine at the top of the S4, which carries positive charges, and attach a fluorescent molecule via the sulfur chemistry. When that part of the channel changes conformation, then we see a deflection in the fluorescent signal. So we can look at the ionic current coming through the channel at the same time that we watch these different parts of the channel move. This is work that Zoltán Varga did, and he was able to characterize all 4 of these constructs and show that we can look at the ionic current at the same time that we can watch the voltage sensing domains move in each of the 4 repeats of the channel. So this was work that Wandy followed up with looking at mixilotine. So we have mixilotine. We want to understand how mixilotine is interacting with the channel. And if you look at domain 1, 2, and 4, we don't really see a shift in the fluorescence dependence on voltage. So as we apply different voltage steps, we can see these deflections in fluorescence that correlate to the movement of the voltage sensing domain in that repeat. And we don't really see much of a change in 1, 2, and 4, but we see a significant change in domain 3, where we have to apply quite negative potentials to start moving the voltage sensing domain back to its resting state. So it's like mixillotine's getting in there and it's jamming the domain 3 voltage sensing domain in the activated conformation, and we have to pull quite hard to get it back down to the resting state. So then we looked at the variants, particularly the 2 that I talked about in one of the first slides. First, we want to understand whether tonic block was consistent with the clinical phenotype. We had to use higher concentrations of drug, and that's because of the setup with the oocytes. There's some access issues, so you need higher concentrations. But we did see something that was very consistent with the ECGs, where you need more drug to block the channel where the patient was resistant, and you need less drug to block the channel for the patient that responded well. Now, what was interesting, and this is different than what the classical theory would suggest, was that there wasn't really a strong dependence on inactivation. The M1652R had the same inactivation as wild type, whereas R1626P was shifted. So that trend wasn't preserved. But what was interesting was that there was a change in the domain 3, the repeat 3 voltage sensing domain activation, where we had a right shift, which means that we needed higher potentials to move the domain 3 to the activated state in M1652R, and R1626P seemed to be in the activated state consistently. And what was surprising is these variants were in D4. So it's like the variants in D4 are affecting the movement in domain 3. So this was exciting, but we couldn't really make a lot of conclusions based on 2 variants. So we looked at all the variants that we could find that we had ECG data for, and we found a very strong correlation between the half potential at which the D3 activates and the tonic block of the drug. So there's a strong relationship between how well mixilotine blocks and the position of the domain 3 voltage sensing domain. So then we wanted to say, okay, can we translate this to what the patients saw? And so we used a partial least squared regression model. First, we did feature selection, and this is just to test which features that we were looking at might be most predictive of the QT response. And we found 2 for the QT interval. Interestingly, it was much harder to predict use-dependent block than it was to predict change in QT interval. So we found 2 features that we needed to predict change in QT interval. That was the domain 3 VSD, V-half, and also this time constant of slow recovery from inactivation. So depending on how the variant affected these 2 features, correlated quite strongly with the patient change in QT interval. So at this point, we were quite excited, and we called the Priori lab and we said, we're ready to publish this paper. And they said, hold on a second, we're actually going to send you new variants, and we're not going to tell you how the patient responded. So put that into your model and then tell us how the patient responded. And so we did that. And we did pretty well. So we got 7 out of 8 within the lower and upper bounds that we predicted. We missed on one, and that patient had a QT interval of 814. That's an extreme prolongation. So 450 is normal, 814 is a new regime of prolongation. So we're not really surprised there. We think something different might be going on with these patients with very long prolongation. So that was interesting. But then we started to think about how we were going to apply this. And we started thinking about whether common variants. So the long QT variants are pretty rare. But what we were wondering is, are there common variants that lots of people have that might respond differently to class 1 drug therapy? And so one is Q1077 deletion, and people tend to have some what we'd consider wild type, some Q1077 deletion. That doesn't make such a difference. But S1103Y, which is highly, you find more in Black patients, those cells tended to have more tonic block and also more use-dependent block. And so that's where we're starting to use the patch liner now, is to look at these different drug blocks. It's a very efficient way for us to look at this, and we can use drug concentrations that are much more reasonable compared to the oocyte system. So you can see here we can measure tonic block, we can apply pulses at 10 hertz and 20 hertz, and we can observe the tonic and use-dependent block of the sodium channel. Then we can look at lidocaine and mixilotine, and you can see here for the tonic block, for a common variant H558R, which is found in about 20% of patients, we see better block of lidocaine. For S1103Y, this is consistent with the oocytes, where we see stronger block from mixilotine. And then when we do the use-dependent protocols, it's kind of interesting that the H558R effect on use-dependent blocks disappears, but the S1103Y effect on mixilotine is actually more strongly pronounced. So we looked at late current, which was kind of interesting because we imagined that the late current block would be different. We didn't really see much of a significant difference, although I will note that in HEK cells, there's not much late current. And so we don't have strong confidence in our ability to measure it in ours or other people's, at least in HEK cells. This is our summary data, where we really saw a strong difference between S1103Y compared to wild type. And we also saw for lidocaine, we saw a difference between H558R and wild type as well. So I think I'll finish with this slide. So what are we doing now? We're actually enrolling patients, and we're looking at their ECG response before and after they get prescribed mixilotine. We're collecting whole exomes in addition to metabolism data, so to understand what different metabolites are in their blood. And then finally, we're collecting IPS or peripheral blood monocytes that we can convert to IPS. And so excitingly, we found 2 patients, one who had H558R and another that had the S1103Y variant. And so we're going to test whether or not these variants in the iPSCs affect the drug response. So I think I'll stop there. I'll skip through this real quick. And I'd say that the planar patch camp has accelerated our ability to interrogate class 1 interaction, and that these genetic variants have a pretty significant effect on antiarrhythmic drug pharmacology. So with that, I'll take any questions. Thank you.
Thanks very much, John. That was a great talk. Really nice to see how well your algorithm predicted the outcome of the drug. Marcus, you have a question?
Yes, thanks very much, John, for the very nice talk. I wanted to ask your opinion about what is so special on D4, because you looked at all the 4 voltage sensors with the patch clamp fluorometry, and you said that only D4 has the biggest effect. What is the, do you know the reason, or why is that so special?
Oh, so why is the drug interacting with D3? That's an interesting question. I don't think we know because it comes into the pore.
Mhm.
And there's some theoretical simulations that say there might be some interaction between the drug and D3, but I don't think that's super clear yet. I know some people have done some simulation studies, but I haven't seen anything super convincing that says, okay, why are we seeing a huge effect on the D3 versus D4? We have some hint that maybe it could be the close state inactivation. But we don't know that for sure
Okay.
yet.
Interesting. I mean, I think for potassium channels, at least there is somehow known this stoichiometry, so that you have also 4 subunits, and then they talk to each other, and then they make a pre-open state, and then the third or the fourth one makes the real open state or so. I don't know. I was just curious if you know something about this kind of stuff, but it's quite biophysical. I have to
Yeah,
say.
so
Yeah.
I think Baron Chanda's done a little bit of work on that, where they look at kind of a defective channel that has inactivation deficient, and then they can see the different voltage sensors move and contribute. But I think the question is did they really change how the channel functioned by introducing that variant?
I see. Okay, thanks for your reply.
Absolutely.
Thanks, Marcus. Thanks, John. I have maybe a couple of questions. With the late current, do you ever enhance it using like ATX or something? Do you have an opinion about?
Oh, that's an interesting idea. So you mean so we can see it better?
Yeah, exactly.
Yeah, that's a good idea. That might help us quite a bit because we really do have a lot of, especially with the HEK cells, we see it more in the IPS cardiomyocytes. And the HEK cells, it's so small. We're not sure that we could see a difference if it was there.
Yeah. I have maybe one other question. Then the cells that you've been using, are they're HEK cells with the mutated channel expressed in them, right? Or
yeah, we transiently transfect them.
okay, and then you use those on the patch liner.
Yeah, that's worked pretty well for us.
Okay, great. So you get a good transfection efficiency and then therefore a good success rate.
Yeah. I don't think we need 100%. We usually get 5 or 6 out of 8. So
Okay, that's
yeah,
pretty.
it's
That
pretty
sounds pretty good for transient transfection.
Yeah,
To my ears.
Yeah, and it would be a lot of work to make the
Stable cell line.
yeah, the stable sub for each variant. So
Yeah.
that ends up working quite well for us.
Yeah. Do you plan to use like patient-derived iPSCs or something?
Yeah. So we have the wild type recordings, and in the next week or two, we should have, because they need to get to day 30. So we've created the iPSCs. They're growing in the lab now, and we've actually edited them. So we took the patients with the S1103Y variant and the H558R, and then we reverted those back to the more common sequence. So we should be able to compare from a patient's cell with and without that variant, whether there's an effect.
Okay, cool. I have one other question here. Mexiletine is known to be pro-arrhythmic. Do you know if it has any other ion channel effects?
I don't know if it's from the physicians we've talked to, they say that it's more of a placebo, which is kind of interesting. Because our thought is that it's probably not working for most people, but there might be a few people, like patients with maybe S1103Y, where it's effective. But we have observed that it probably interacts with the hERG channel, and that interaction might be beta subunit dependent. So that's something that we're looking at. It's kind of interesting. We always think of the beta subunits as just interacting with the sodium channels, but it seems like there's something, at least when we knock it out of the iPSCs, we start seeing a K-channel interaction.
Okay, interesting. Okay, well, I think that then just remains for me to thank both of our speakers, to Sam Salvage and John Silva, for these 2 brilliant talks. Really found them super interesting. And Jamar, do you have any closing words for us?
Yes. Just 1 second. Yeah, so also just besides thanking Samantha and Johnathan, it was really nice talks. I would like just to give you really brief some information then. So just remember that we have the SynchroPatch instrument grant. The applications will open really soon. So you will be able to download the form and then send us the application until the 20th of October. Also, we have coming up our Nanion user meetings for what is the rest of this year. So we will have in October the Nanion user meeting in Munich in the headquarters. And later on in November, we will also have the user meeting in Japan. So the registrations for the user meeting in HQ are already open, and the ones for Japan will open next week. So stay tuned for that. So these two events will be also in a hybrid format. So you will be able to join from your desk at home or at the office. Yeah, and then concerning the events that we are normally hosting, is just an invitation for you to go to our website to the news section so that you can stay up to date with all the events that are coming up and all the activities that we are preparing for you. So yeah, without further ado, then I would like to thank you all for joining us and yeah, for being part of the discussion. And it would be then until the next occasion that we will see each other. Okay, thank you.